Thank you very much for introduction. Again, my name is Nikolai Kalugin. I'm professor of materials engineering at New Mexico Tech. And my today's story will be about engineering carbons from biomedical applications. So we work on carbons and graphene for many years, and our beginning many years ago was purely scientific, but at a certain point we realized that it's about the time to transfer all this tremendous potential of engineered carbons to different applications. And looking on the landscape and the actual activities around the globe, so we decided that for us it's probably the best niche for us would be biomedical research with carbons. So us, I mean, who are us? It's me, it's several of my current and former students, especially Lindsay Candelari, who did Ph.D., defended Ph.D. successfully a little bit more than a year ago. And Dr. Lilia Frolova, Alexey Serov from Pakharita Powder. We collaborate with Pakharita Powder very actively and very successfully. Peter Kalugin from Harvard. And I would like to appreciate the support from New Mexico Tech Research Foundation. So again, carbons and what other people did with carbons, how graphene started. It was, high-speed electronics, interesting quantum phenomena. But what is also interesting about carbons is normally carbons are very chemically stable and very chemically inert because of a particular type of bonding between atoms. It's usually the standard of chemical stability, and therefore it's a standard for fantastic biocompatibility. It's one of the most biocompatible material. It's one starting point. The second, again, chemical stability usually means that it's not that easy to do something with and at the surface of carbon. So if you would like to modify the surface somehow, it's a very challenging thing, and still many things were demonstrated, but majority of them, sorry for technical details, majority of these modifications are not covalent. So it's easier to attach something, but it's similarly easier to detach something and to lose the functionality if the conditions are anyhow aggressive. So we found a solution in this problem. We particularly explored several reactions, which link also carbon-containing molecules to surfaces covalently, and it opened up the wide spectrum of applications and capabilities. One of them, at the left side of this slide, is the illustration of how carbons, in this case, again, we are talking about sp2 carbons, so everything carbon made except diamond, because diamond is different allotropic form of carbon. It's sp3 hybridization. It's a whole different story chemistry wise. So we are talking about graphene, graphite, carbon nanotubes, and all other conductive allotropic forms of carbon. So at the left side, one of applications we recently demonstrated, it's a use of sponges carbon for separation of chiral or what is chirality? It's a spirality element and symmetry of molecules. So chiral drugs were separated successfully using our materials. We demonstrated alternative, and better, we believe, we not just believe, we know for sure, we make better separation columns than the commercially available things. And also, I'm already moving to the right side of this slide. We also developed the techniques for decoration of carbons, not necessarily sponges carbons. It may be really uniform, structurally uniform, and very conductive carbons. So decoration of these conductive materials with different important biological molecules, including peptides, including antibodies, or we know actually how to make a so-called click chemistry on the surface of carbon. What is basically click chemistry, just in one word, is exactly the same idea which our own immune system uses. So if we have, for example, attack with pathogenic organisms like viruses or bacteria, our immune system recognizes the molecular fragments on the surface and responds in generation of antibodies. It's Y capital letter shaped molecules which link to active parts of invader and passivated or make these points of connection as a sort of alarm flags to immune system. So the affinity between the molecules on the pathogen, indicating the presence of pathogen, and the corresponding antibody. It's one of the mysteries of this life. It's very, very addressed, very, very selective. So we decided also to use this literally log and K mutual compatibility of components for assembling things at the surface of carbon. Idea is to make literally clickable smart electrodes to, for example, nervous systems or to other parts of biological objects, on demand, without any use of complicated, super precise, super sensitive XYZ 3D positioning techniques. So we make electrode, we make carbon, we decorate it with necessary chemistry, we place it at the right place in the organism, and it clicks in a smart way, in a self-organized way, exactly to the particular place at the cellular membrane where we would like it to be attached. So these are the two major ideas, and for this presentation. Okay, what is now? Okay, great. So as I said, carbon separation materials, if you're talking about carbon separation materials, the key points, it's enhanced chemical stability. It's a much lower price for fabrication compared to the current solution for separation techniques, which is usually pretty complicated. Silica or cellites like based matrices, also decorated with carbon molecules. It's also, as I just mentioned, it's good. We know how to bind biologically significant molecules like potentially DNAs, amino acids, sugars, and whatsoever. And when we are talking about these materials for electrodes, they are biocompatible, they are not destructive, and they are highly specific. So again, speaking about carbon surfaces in a more, if you look at the bigger picture, so you can really, using our technology and using our principles, you can do many different things. You can make new type of electrodes to living cells. You can use these materials for different types of affinity chromatography, so specifically separation techniques for different important biological molecules and compounds. It may be electronic non-optical DNA sequencing, solutions, biosensors, scaffolds for tissue engineering, substrates for synthesis of important peptides, and last but not least, self-organized fabrication of carbon-based electronics and optoelectronic material cell devices. Basically, this is pretty much unexplored direction so far. So speaking about potential of market for separation of drug isomers, it's important to mention that majority of important compounds around us, important drugs, are actually chiral. Our own organism is also chiral. We have one particular orientation, one particular variant of spirality of our DNA, and of all other proteins, peptides, and other biologically significant molecules in our organism. So when we use drugs, if you synthesize these drugs without chiral separations, then about 50% of mass and volume of material we generate is, in the best case, useless. And this is the reason why brand name, for example, antibiotics or anti-inflammatory drugs are so expensive, and why generics are cheap, but because generics are typically not chirally pure, and it's typically 50% of mass you buy, 50% of chemical you buy is actually useless for your organism. So chiral separation is important, and it's a big pharma market with all these multi-billion numbers. Speaking about applications of, in the field of electrons and connections between electronics and neurons, this field is just developing, and already still the global brain implants market size, for example, is valued at the level of about 4.6 billions in 2020. Again, it speaks for itself. People like Elon Musk invests a lot into these interfaces. His Neuralink project is exactly, the company working on development of the new generation of of interface between electronics and nervous system. Issues, there are many issues. It has to be non-invasive. It has to be not triggering the immune response. Excuse me?
One too many.
Okay, I thought it's a question. Okay, so potential market is significant. And what we demonstrated at the left side of this slide, it's, for example, the demonstration of successful separation of thalidomide. We use it a commercial, not purified thalidomide, and we demonstrate the baseline separation into two enantiomeric fractions with 100% separation. Why thalidomide, for example, is important? You may remember the crisis of 1960s, 1970s, when thalidomide was just developed and introduced to the market, and many young mothers, during pregnancy, many women used it as a motion sickness cure, and then the wrong, opposite enantiomer of the drug was causing many birth defects and very severe consequences. So later on, people realized that the reason of these problems is exactly, not sufficient purity or no chiral purity of materials. So, for example, with our material, we can solve this problem. It's one thing. So at the right side, it's a demonstration of the performance of our electrodes decorated with different peptides for addressed linking of these electrodes with opposite parts of antibody-antigen chemical reaction. And instead of neurons, in these experiments, we used quantum dots decorated with corresponding antibodies, commercial quantum dots. So under light, quantum dots were decorating the pulse of electrical polarization, and you see the spikes, the red curves. We clearly see very high selectivity of our electrodes to particular counterpart, and also sufficient sensitivity, so we detect even these test signals. So you see the sensitivity at the level of 50 microvolts is achieved. Transmembrane potential, when our own nervous cells work and generate signals. So it's about 2, if not 3 orders of magnitude high signals. So we are definitely, sensitivity-wise, we are in a very good shape. So
Dr. Clune, so just to step in real quick, I know you have to leave kind of early, and so just want to make sure we get a chance for our
yeah, this is my slide.
shark surgeon.
So yeah, I just demonstrated a couple of illustrations of what we do. This is not the only, these are just the biomed directions. All together, we have at the moment already pretty solid portfolio of different applications. And you may see in this slide what we do and what current, provisional or already granted patents we have about carbon-related technologies. So I will finish at this moment. So thank you very much. And questions?
Thank you. All right, over to our sharks. And really quickly, just want to thank everybody, Matthias Weber from Mighty Capital, John Chavez from the Startup Factory. And so, yeah, this has been a great shark session. So thank you guys for participating. And yeah, I will hand it over to you.
I have a question, if I may start very quickly. So you showed several applications, some more concrete than others. How or which applications do you believe have a potential to bring to market? And if so, how and by when?
Chiral separation needs just, formalization of things, creation of company, or, negotiations and decision about how to do it. But we have a fully developed technology, and yeah, we also know how to scale it up. So this part is, the things were published already more than 2 years ago. That's, yeah, so it's ready to go. It works, it's there. The remaining technical things are purely engineering. It's about how to make the packing of columns, really stable, and it may need some iterations, but it's really good. About electrodes, it's different. It's a very innovative approach, really innovative approach. I'm not exaggerating. I mean, nobody did it like this before. And where we are at the moment, we need to move from purely, on-table experiments to a real laboratory where we may apply these electrodes to mice. So it has to be a biolaboratory where they have interest, culture, and ability to deal with, real interfaces. I mean, it will show probably new problems and limiting factors, but this is the way to go.
Okay, and maybe a follow-up question on the first one, the separation of materials and isomers.
Mm-hmm.
Do you plan to license your technology out, or will it likely result in a spin-off? What is your thinking?
We should sit down and discuss. Both options are sounding interesting to us. Yeah, and of course it's a property of New Mexico Tech and Rainforest. So yes, these guys should also participate in the discussion. But both variants are interesting. Yeah.
Okay.
Definitely.
All right, great. Sharks, any last questions before we adjourn for lunch break? All right. Well, thank you to all of our speakers. Thanks for everyone for coming and joining us this morning. We're going to take a break now until 12:45. And in the meantime, if you are eating at your desk, we have some company or some partnership highlights to all of our partner institutions that are here today that you can watch while you eat. So look forward to seeing everybody after lunch, and we'll talk to you all soon. Thanks.